Hello everyone. So thank you very much for inviting me today to present some of my work. So I will try to present you, instead of looking at specific part of my recent work, I will try to more present you another view of my different research during my career and try to illustrate you something new, hopefully for you. And yeah, anyway, I want to show you first that my career, my research is divided in two main directions, two main kind of research. Pure basic research, where I'm trying to reproduce human genetic disease in small animal models, such as sea lichens and zebrafish, models I will present you today. And then use these models to really understand what's happening in this human genetic disease. And another part of my research is much more focused on drug discovery, okay, obviously just finding drugs for patients. But I really want to illustrate you during this presentation that actually these both directions are strongly interconnected, because obviously I cannot do good drug discovery without good model of disease and compatible with large-scale screen. And as well that actually my drug discovery part really helped me to understand better the human disease I'm working on. And this is what I call chemical genetics, using small molecules to investigate specific disease, to investigate biological system. And I will try at the beginning of this talk to illustrate you that a little bit, because I think it's underused in our community, this chemical genetics approach. So I really, even if it's today, it's just a very, not a big part in my work, it's really something powerful. And I will try to illustrate you that. So let's go in the talk. So yeah, very briefly, I will introduce chemical genetics by showing you some examples. And after, I will very briefly introduce the zebrafish model, because I believe some of you don't have much experience with that. So a brief introduction, because all the rest of the talk will be based on zebrafish. And then, yeah, I will show you how I developed new tools for zebrafish that really open new avenue for disease modeling and large-scale screening and creating models for large-scale screen. But I will more illustrate you the power of these tools, and that has a real impact probably in your research. But as well, I will show you how I developed these tools and how I use these tools to reproduce for the first time in zebrafish the different form of the motor neuron disease, spinal muscular atrophy. Okay, I will show you the interest of these models, which are fantastic for drug discovery and both understand this disease. And then I will illustrate you, I will show you the versatility, I will demonstrate the versatility of these tools by showing you some experiment I made using these tools to respond to specific questions that belong to SMA, to the biology of SMA, and I will show you that this was not possible before, and using these tools we can now really respond to new questions using the zebrafish. And I will then, I will then show you what we are now trying to initiate more in terms of drug discovery and chemical genetics using SMA. And finally, I will show you that what we are doing on ALS, amyotrophic lateral sclerosis, which is another MND, what actually we call motor neuron disease here in Australia. And I will show you that we are applying kind of the same principle to ALS. I will briefly talk about that, because it's also something we have a good result right now with. Anyway, so let's go in the proof of principle. So to illustrate you a bit the power of chemical genetics, I need to go back at the beginning of my career. So at the beginning of my career, I was working in Sanofi Aventis in a pharmaceutical company in Europe, and the idea was to set up large-scale screen, to set up high-throughput screening to find drugs, in vitro high-throughput screening, to find drugs that would bind or modify the activity of a specific target, a target known to be involved in a disease. At that time was Alzheimer, Parkinson, and a few others. And then finding such compounds, we were trying to climb the ladder of a traditional drug discovery pipeline to find, to develop a drug for human. And they were involving different departments. So by looking at that pipeline, that traditional drug discovery pipeline, you may understand that if you don't have a good target to start with, it's very hard to develop a good drug for humans. And if you don't have target, and unfortunately, most neurological disease and neuromuscular disease, the disease I'm working on, are poorly understood. We don't understand them well, so we don't have good targets, and even for some, we don't have targets at all. So this whole pipeline is not working well. And if we take the example of MND, of motor neuron disease, it's even worse because these diseases are considered as not bankable, meaning the industry doesn't feel that these diseases are value for money for them. So they don't put a lot of effort in trying to identify targets and establish these screens. They wait us, the academic system, to bring these targets on the table to then initiate the screen or to bring compounds already developed. So yeah, so this pipeline is kind of useless until you don't have a good knowledge of your disease. So at that time, I was then very attracted by another approach which was emerging in the academy, in the university, in the public system, sorry, which was the idea was to use small animal models and phenotypic screening, meaning trying to reproduce your disease in animals such as ciliegans and zebrafish that are compatible with large-scale screen, and try to get a phenotype that is relevant to human. And then, instead of looking at a target, you look at the phenotype, and you look for compounds that reverse this phenotype, all right? And so that's what my idea. I wanted to go in the university to do a PhD to demonstrate that it was indeed feasible and try after to sell that to the industry at the World Department. But I was also at the same time very attracted by basic research, by actually understanding what I was doing, understanding the disease I was working on. And by looking at that, I said, look, if I'm able to find a compound here that is able to reverse my phenotype or aggravate it, and if I'm able to identify the mechanism of action, the target of these drugs, I should be able to quickly pinpoint new entities, new targets that will feed the traditional drug discovery pipeline, and at the same time, I will be able to use these targets, these probably unsuspected targets, to better understand the disease I'm working on, which I call chemical genetics, using these compounds to quickly identify targets through another approach, something that is not really used in the laboratory right now, and using then this target to understand your disease. It's similar to a forward genetic screen, but using chemicals. So let me show you an example of that. So when I started my PhD, I worked on different models, including one which, in ciliegans, a model of Duchenne muscular dystrophy. So it's nothing to do with the brain, but it's a muscular disease, and it's a very good example. So this model was showing a phenotype very similar to humans. So when you take out the gene dystrophin in the worm, which is the gene responsible for this disease, a muscular dystrophy, right? So when you take out this gene, the worm starts to have a progressive muscular degeneration, as in human, and a progressive loss of motor functions. And interestingly, this worm is perfect for establishing screen and testing drugs. Why? Because he has a defined number of muscle cells, 95 muscle cells. You can see here 4 muscle cells, which are diamond-shaped muscle cells in this worm. Okay, so they do not fuse, and they are in a number 95. So that means that, and they cannot regenerate. So when they degenerate, you will see them like hole like this in the muscles, and you can directly count this number of absent muscle cells, which will be the perfect reflect of the progression of the disease. For example, this one has a score of 0, it's just healthy, and this one has a score of 4, and this number allows you to compare different treatment. So that was a very good model to start with, and great to establish screens, but you might understand that this is not really compatible with large-scale screen, and we had to engineer this to make it compatible with what we do in industry and to try to after sell that to the industry. So let's, for example, to quantify these muscle cells, you need to collect this animal, to wash them, to do a permeabilization of the cuticle, to do a phalloidine staining, which is actually quite expensive, by the way, in a large number. And after put this worm under a slide and each population, and then count manually each worm under a microscope. So that's feasible, but it's not really compatible. That's feasible for a small pool of compounds, but not compatible with a large pool of compounds. So we had to find a way to make it more automatic. So what we did, we expressed GFP in the nuclei of the muscle cells, and as these muscle cells do not fuse as our muscle cells, they remain, they only have one nucleus. So then you can see in this worm that we obtain around 95 nuclei fluorescent dot, 95 fluorescent dot, reflecting the 95 muscle cells. So we had to do nothing but just look at this worm in a dish to evaluate the number of muscle cells in this worm, to qualitatively assess the musculature of the worm. You can see here absence of fluorescent dot reflecting muscular degeneration. But we went further than that, and we used a cell sorter, kind of a cell sorter for worm, that was able here to evaluate these fluorescent dots and give numerical values that you can use to automatically compare your different treatments. So by establishing such a screen, we tested around 10,000 compounds. So we found a number of hits, and we validated 26 of them in mouse model. So first, that was a big demonstration. Yes, you can use small animal models to find drugs that come back and that present an alternative to the traditional drug discovery pipeline. Okay, that's very powerful. But at the same time, so I wanted to show that that could be also very useful more in terms of basic research, and that we can use that to identify unsuspected targets in our disease. So, but unfortunately here, I was surprised because I was a bit fooled. Understanding the function of even one drug can take longer than a PhD. It can be very complicated when you have no clue about how your drug works. So that was a big deal. But let's wait. Actually, so I told you that here we obtained 26 hits, and actually a bit more. But when we looked carefully at these hits, we find that there were only three main chemicals families, three main chemical scaffold in this pool, and that were belonging to sulfonamide compound, tricyclic compound, and benzodiazepine compound. And we know that there is approved drugs in the market belonging to this scaffold, and with known mechanisms of actions, we know few targets of these drugs. So let's take the example of sulfonamide compound. We know that they are strong inhibitors of the enzyme carbonic anhydrase. And there is, for example, two known drugs available in the market called naptazane for metazolamide and daranide for dichlorphenamide. So we tested these drugs in the worm, and as no surprise, they worked quite well. So you have here wild type with this 4 muscle cell, here the DMD with this absent muscle cell. So you can see here that this animal presents 6 absent muscle cells per side of worm. And when you treat this animal with dichlorphenamide, daranide, you nearly blocked completely muscular degeneration, and you also restore motor function. Only 1 absent muscle cell in this worm. And then, as we know that they are inhibitor of carbonic anhydrase, I looked into the worm to the available carbonic anhydrase. First, as the siligance presents some carbonic anhydrase. And yes, we find 6 different. So we test them individually in the worm, and we found that actually downregulation of CEH4 was sufficient to block or slow down muscular degeneration. And we showed later that actually CEH4 was indeed the target of dichlorphenamide, and they were acting synergistically in reduction of muscular degeneration. So very powerful, right? So we find a really unsuspected target here involved in muscular degeneration that would probably not be found easily through another approach. But we went further than that, meaning when I looked at the expression of these different carbonic anhydrase, I realized, and I saw that demonstrated that CEH4 was the only carbonic anhydrase expressed in the mitochondria of muscles. And this helped me to really decipher and to point out my research to these specific organelles. And this helped me to find additional compounds that block muscular degeneration that were not identified during the screens, and additional targets, player of muscular degeneration, additional targets that we can use to try to treat patients. So briefly, we went with a model. I will not detail that. It's not the point today. But just to show you that chemical genetics approach, the drug discovery approach, allowed me to identify all new mechanisms in muscular degeneration and new drugs and new targets that involve mitochondrial stress, mitochondrial fission, and release of apoptotic factor, and so on. So that was very, very powerful. And just also to illustrate you the power of chemical genetics, when I started this research, this PhD, my pure wish was really to demonstrate it was feasible and after to sell that as a kind of a technical approach, a platform to the industry. But when after in PhD, I really fall in love with basic research, which I mostly do today. And I wanted then to try to make a career in the academic. But my supervisor at that time was a bit stressed out because he said, I'm not sure it's a good idea because the drug discovery approach will never lead to good papers. I guess he's kind of right, but if you use this wise approach, if you use specific chemicals, as I will show you later, actually it can be very profitable for someone that do basic research and by doing chemical genetics. And just to illustrate you for DMD, just for this PhD, we obtained, related to this PhD, we obtained 3 patents and 5 papers starting from compounds and describing new mechanisms. So it can be profitable. So today, after that, yeah, I decided to continue in postdoc and kind of focus more on basic research, more understanding what's happening in this kind of disease, especially neurological and neuromuscular disease. But I still try to use my chemical genetics approach when I can. And I focus my work. So yeah, obviously I try to still model. I will show you. I try to reproduce human disease in different small animal models and try to understand them, but I'm now also trying to use my chemical genetics approach. And I say chemical genetics and not drug discovery, because I initially use a very targeted and kind of a method where I use very specific compounds, including the low-pack chemical library, press-quick and spectrochemical library, because this library are enriched in compounds that have known mechanisms of actions, okay? We know their targets, so you can easily and quickly, if you find some that aggravate or improve your phenotype, you can quickly jump into your targets, and that will help like a forward genetic screen, right? And you can quickly investigate, quickly pinpoint mechanisms in your models. So I'm starting to repeat myself a bit. But yeah, that's very powerful, and that's kind of unique. I think it's a kind of a niche here that has to be developed more. But also, anyway, for patients, meaning, for example, the press-quick is also enriched in FDA-approved drugs that are already approved. So if we find drugs that here rescue our phenotype, we should be able to quickly partner up with a company to try to develop a drug that would be quickly available for human because it's approved and we can easily go for a specific dose in clinical trial. This is what I'm trying to do now. So that's it for this demonstration of the power of chemical genetics. Hopefully I convince you a bit that it can be powerful. So now I want to introduce the Zebrafish model because I will talk about this model up to the end of the talk. So this model was introduced into the laboratory as silicons to study the development. And for kind of the same reason, it's just relatively easy to maintain and not too expensive to grow. But most importantly, you can produce a large number of eggs every day, a large number of individuals. So you can see here male and female giving these eggs. You can see me collecting these eggs. And these large numbers of eggs you can produce every day, they develop externally. So you don't have, for example, to open the mouth to get the embryo. And you can see that in one day, you can go from one cell to a fully developed embryo. So, and in 3 days, you have a fully developed larvae with a complex vascular system. You can see the blood going through, complex neuromuscular system as well, which is important for my research, developed brain and functional brain, optical system, heart, etc. So I'm sure you understand here by looking at that how powerful this model is to study the development. However, it's also very attractive for medical research and drug discovery in my field. For kind of the same reason, but also because it's transparent. You can see before the muscles, now the motor ones. And obviously, this is in vivo, in a live fish, so you don't have to sacrifice this animal to look inside. And and I can track in my models, I can track the degeneration of this tissue in a motor neuron disease model, for example. When a motor neuron degenerate, I can see it in vivo in real time and analyze and record what's happening. So it's very powerful. But they are also compatible with multi-well plate, like you see here, with one larvae per well. So these wells are that big. And yeah, I developed different assays with also my colleagues that allow us to easily test different drugs and to also analyze the motor function of these fish, etc., etc. I'll not go in detail into that. Let's focus on that. So now I will show you some new tools. I will demonstrate you the development of new tools optimized and developed for zebrafish. That really new tools that really fill the gap in the zebrafish genetic toolbox, and that really fill the gap between morpho and CRISPR, and something that, for example, that allow, I will show you later, tissue-specific downregulation of gene of interest, but also you can now downregulate any gene you want through the whole life of the fish. Which was not possible before. And I will show you how I use these tools to develop the first table to reproduce a different form of SMA in the fish. So, briefly for the little history, is when after my PhD, I started a postdoc, and I had a strong interest in different diseases, including SMA for spinal muscular atrophy, which is a disease linked to SMN1 for survival of motor neuron 1. So this is a gene ubiquitously expressed in all your cells. But no one of your cells can survive without the protein SMN. Okay? So it's expressed in all your cells, but no one of your cells can survive without this protein. However, if you have a low level of this protein, you will develop the disease. And lower the level in your body, stronger the disease will be. We classify this disease in 4 different types, from type 1 to type 4, with type 1 the first cause of infant death in terms of genetic disease in the world. And type 4 that only present a late onset form, only when you be adult. And what will happen? You will present a motor neuron degeneration of your motor neurons, and muscular atrophy, and obviously premature death. So we don't understand well this disease, and we need better treatments. Even if recently, actually very recently, we start to have compounds reaching the patients. So that's really, really great. Actually, that's a very good history. But we still need to better understand the disease, and we still need better treatment. So, but at that time, nothing was available. And so I wanted to create a zebrafish model of the disease for both understanding and finding drugs against this disease. So I was thinking it would be very easy, because looking at that, I said, oh, and coming from cells and silicons, I said I would just do stable knockdown of SMN1 in the fish, and I will be able to reproduce this different background, having different low level of SMN in the body of this fish. But I was a bit kind of stupid when I started my postdoc and draft this project, even if it was kind of accepted, but is so that actually RNAi gene silencing, knockdown, transgenic knockdown doesn't exist in zebrafish, or it doesn't work. Few tried, but it just doesn't work. So my whole project kind of fall apart, right? So, and just for the history, so you might tell me, so why you are not doing knockout of SMN1? So first, CRISPR was not available at that time. And second, if you understood what I said, if you take out SMN1 completely, if you have no more of the protein, a pure knockout, it's lethal, and it's not reflecting the disease. So you don't want that. So you want to be able to create a low level of the protein during the whole life of the fish. And RNAi was not available at that time. So the whole project kind of was going in a dead end, but before stopping it and working on the other disease, said, let's wait. Why RNAi doesn't work in the fish? So I went through the literature, a few experiments. And here I will illustrate you what we do in cells or in silicons and even Drosophila, I believe. So for example, in silicons, when we want to knock out a gene, for example, SMN1, we give them as food a long double-strand RNA with one strand complementary to your messenger. And this would be recognized by dicer, cut in small small RNA, okay, of 21 to 23 nucleotide long. and the complementary sequence will bind to your open reading frame of your messenger and will knock it down. So this will be destruct, and you will even have an amplification loop because you will have the release of small piece of this messenger that will amplify the mechanisms, and you will have a strong knockdown. So why this is not working in zebrafish? So actually, if so, let me show you that. So if you introduce a long double strand, or if you inject a long double strand RNA in zebrafish, you will induce what we call the interferon response, which is a kind of a immune response, and you will lead to this will lead to a global mRNA knockdown. All of your mRNA will be degraded. So you don't want that; it's toxic. Oh, and by the way, it's also the same, I think, in every vertebrate. So then what about injecting or introducing small RNA, scRNA? So in theory it's possible, but you take the risk to flout the macroRNA, the endogenous macroRNA processing system, and in fish you definitely don't want to mess up with the macroRNA called macroRNA 430. So I will not go in detail on that, just ask me a question at the end if you want. There's a lot to say about that, but anyway, you don't want to mess up with that because you will create also a lot of off-specificity. But even if you were happy to deal with that, for example, co-inject this macroRNA 430 to compensate for its loss, or deal with the off-specific effect, actually in fish and in all I think all vertebrates I read about or look about, if you target the open reading frame, 9 times on 10, it just doesn't work. That doesn't lead to a knockdown as it is in cell or ciliates. So why is that? So anyway, my whole project was gone. But let's wait. I talk about macroRNA here. What is a macroRNA? I said, okay, what is a macroRNA? So here, so you have a traditional gene expression from the DNA to a messenger to a protein, and the macroRNA, the endogenous macroRNA, as gene expressed from your DNA, and they are expressed in mature macroRNA to repress, to control the level of the messenger, meaning the cell use his endogenous macroRNA to knock down his own gene. So this is the endogenous knockdown tools of the cells, and this works in all organisms. So I said, and in zebrafish and in every vertebrate. So looking at that, I said, if a cell can do it, I should be able to do it as well. And this is exactly what we did. Luckily, actually, there was few tools available in the market and in the literature, so we tested a bunch of them, and we created as well some, and we finished with an optimized one for zebrafish. I will not detail this process, just go to the endpoint. So this is exactly what we do now. We are now able to transgenically express synthetic macroRNA to repress any gene you want, and we can repress several genes at the same time. We'll show that maybe later. But how it works in real life, so what we do, we are creating transgene with a promoter you want, so you can use a tissue-specific promoter, but here, ubiquitous expression of our cassette here that will express DS red. So the fish or the cell expressing your transgene will be red, so you can more track them thanks to the color. And then expression of your mature macroRNA of choice based on the design. If you follow the paper, everything is right. It's explained how to design that, and then this will lead to a specific macroRNA. And we showed, for example, for SMN1, that indeed, if we express a macroRNA, which is equivalent of scRNA, targeting the open reading frame of SMN1 in the fish. So here is transgenic experiment, okay? Stable, no injection, nothing, it's stable. It's really unique. If we target the open reading frame, it does not lead to knockdown as kind of expected. So here, the quantification of the protein in transgenic fish with a control, and here no difference when you target the open reading frame. However, when we target the 3' here, we showed that we were able to achieve strong knockdown in this transgenic fish, okay? And we were able to create different transgenic lines, independent lines, showing different level of knockdown, with one line showing up to 90% reduction in SMN1, all right? So first, it was a big achievement, and that these tools now really open new avenue to study gene functions in zebrafish, but I will show you later as well that really they open new avenue, especially in terms of large-scale screen and on terms of pure screening. But anyway, so really a big development. So I'm really now filling the gap between CRISPR and Morpholino. But the next question is, was it enough? So we created, as in patients, different lines with different level of SMN. So was that enough to reproduce a disease? And so before showing the results, I need to briefly introduce the motor neuron in the fish. So you have here 5-DPF larvae with these motor neurons in the spine. So you have the cell body here. And I want you to focus on the CAP motor neurons, which are motor neurons sending a long axon ventrally here that go all around the muscles, come back here, and join what we call the choice point. We want to look at this axon because they are long first and prone to defects, so we can easily observe these defects and quantify them. And yeah, and they're also, there is no other track going through, so they're just easy to analyze. And also prone to defect, sorry, and easy to show you. So here, a wild type, a control, with all the at 52 hours per fertilization, 52 hours after birth. You can see that most of this axon just reaches choice point. However, when we have a downregulation of SMN1, a transgenic knockdown of 70%, we start to have strong defects. Most of this axon present a slow growth, a slow retardation in their development. But they are also heterogeneous in their development. Some are shorter than others, some are hyper-branched. Some even send some axon to try to rescue some territories that are not innervated at all. You can see these axons that just go through the muscles instead of going to around the muscles to reach a choice point. And when we went up to 90% reduction, it was just a mess. Some axons don't project at all. Some send huge branch to try to rescue these territories. And some axons even fail to project in the right path. It's like you will have a nerve that should go in your left leg, go during the development in your right leg. So I'm sure you can imagine how these fish move after that, and I will actually show you. And yeah, and these fish also die very, very early on, at around day 10, the 10 days after birth. And we demonstrated, as expected, that lower the protein, lower SMN1 in the body of this fish, stronger the phenotype was, exactly as in patients. So we were able to reproduce a different type of the disease, including the late onset, so you have here fish which has 70% reduction in SMN1 that had a not strong phenotype when they were early in life. So here a control that moved quite smoothly. Whereas this one, you can see that they start to have a progressive paralysis, and we demonstrated that this fish, this phenotype, is linked to degeneration in their motor neurons, progressive degeneration of their motor neurons, and then muscular atrophy. And they develop scoliosis. We also come back to that later. And you can see that they present strong paralysis. For example, this one has strong paralysis of his left trunk. Just for the quick history, that you can see that these fish are glowing red. They should be actually, a white type is more like dark blue. And they are glowing red because they express the transgene so strongly, you don't need a fluorescent light to see this fluorescent red expression. Yes. Anyway. So very interesting, but yeah, why this video is not showing up? Anyway, and in the strong form of the disease, the fish that have 90% reduction in SMN1, we were able also to observe in vivo and in real time motor neuron degeneration, which is unique, and we can now study this phenomena in vivo. So this is a live fish time lapse where we look at the fish under a microscope for 1 day, so this is 24 hours, okay, a time lapse. So you can, I don't know why this video doesn't work, but you can see these neurons dying, okay, where there's like popcorn, and you can see nerves, just nerve tracks which disappear. So this is very, very quick observation. But so we are now actually working on that to try to investigate and describe what's happening in this degenerative process in the severe form. And that's also very powerful things that you can do with the fish, is the optical transparency allow you to look at these neurons in vivo in real time. But at the same time, at the end of this experiment, you can also get back your fish and continue your analysis, which, for example, is the case. Because so this one has 90% reduction in SMN. They present this phenotype, and I'm sure you understand that they move quite badly. And what we do, for example, here we put them in a dish, in a multi-well plate, 24 multi-well plate, with here controls, and here's these fish that were presenting this motor neuron degeneration. And you can clearly see that these fish move way worse than the control. And here the numerical value, basic treatments, you can definitely see that using this, so here we use a specific device that automatically track and plot that tracks the swimming track and their speed. And you can definitely see that just using these two parameters, we are able to easily assess the disease state. And we can easily try to find drugs that would reverse, or meaning make it better, improve the motility of this fish, and try to find drugs that reverse and cure, that is beneficial for SMA. That's actually one of the best models from my experience, one of the best zebrafish models I've saw, to establish large-scale screen. And then we come back to that later. But on top of that, sometimes some people, a lot of people in our field talk about large-scale screen, drug screening using zebrafish or sea ligands. But when it comes to make them, they realize it's not that easy, especially when you work on the disease, because it can be the result can be very heterogeneous, right? And more complicated is your readout. More complicated is your readout is what you are looking at the end to see if your compound has an effect. So more complicated is your readout. More complicated is the treatment of the data, and you will generate a lot, or that will increase the number of false positive and false negative in your screen. However, if you have a screen which is a binary screen, meaning the endpoint, the readout, what you are looking at is just 0 or 1, it's much easier and much quicker. And here, for example, all of these fish will die between day 10 and day 11, and this can be used to establish a binary screen. If you put yourself here at day 12 and look which well you have a movement, which well you have a fish that is alive, then you can screen thousands of compounds in a very short time frame, because your score is just dead or alive, 0 or 1. And doing that, you can be very quick in your screening. Anyway, so that's a very good model, and we try to use these two readouts, dead or alive and also motor function. And we come back to that a bit later for this drug discovery. And now I just want also to illustrate you the power of these new tools are created to manipulate gene functions, and how many actually maybe you can use that in your own research to respond to specific questions, things you were not be able to do before. So just to introduce that. So for example, for SMA, there is several questions obviously that we want to answer, and there is one. The main question is, as I said before, considering that this gene is expressed in all your cells, okay, and no one of your cells can survive without the protein. If the protein is gone, no cell can survive. No cell can survive without SMN. So why, as it expressed everywhere, a low level of this protein lead to specific phenotype in your spine and in your muscle? So behind this question, a more medical question is, do you need to compensate for this loss in your whole body? Or can you try to develop a treatment or an approach to focus only on your spine, to compensate for this loss only on your spine, or only in your muscles, or in both tissue, or actually in upstream circuit in your nervous system? So that's big questions. And this is, so to try to respond to this question, what I did is I created tissue-specific model. So instead of expressing ubiquitously my microRNA, I express this microRNA only in motor neurons or only in muscles and also in other tissue. But let's focus here on motor neurons and muscles. So I use a GEL4-US system. I will not go back too much in that. So that allows you to obviously express your cassette tissue specifically. Ask me a question about that if you want. I will just come back very briefly to the GEL4-US system a little bit later. But so here we have very similar approach, very similar cassette with DS-RED. So you can track, be sure that your motor neurons are knocked down by following with red. But we chain, to make it more potent, we chain 4 different microRNA. Just for that, just to illustrate that, is why we chain 4 different microRNA. So yeah, to improve the potency, but we wanted to do that because initially when we developed this model, it was probably a bit lucky, by the way, but it was difficult to identify a line showing a very strong knockdown. Most of the line we generated were in the range between 70% 65, 75% reduction in SMN. And we were lucky enough, we work hard for that, but to get one line presenting 90% reduction. And to then limit the time we had to spend on screening a large number of fish, we chained that, and then it works very well now. It just leads to strong knockdown without having to screen a lot of line and find the good one. Anyway, so we created tissue-specific knockdown of SMN1 like this. Here's the results. So you can see here the control, which as previously shown at 52 hour post-fertilization, most of the CAP axon just reaches choice point here. In the muscular knockdown fish, we did not detect any significant defects. Okay, so all fine, nearly all fine. Well, when we downregulate in motor neurons, we were able to reproduce most of the phenotype observed in the ubiquitous line, and including short axon, path-finding errors, hyperbranching. Okay, so here an axon that did not project at all, like with this new projection here that got here to the choice point, like it's a mess, like a very short one as well here, and also motor neuron degeneration. And we also were able to reproduce late onset form of the disease with the motor neuron knockdown. So here wild type, here muscle knockdown, where these fish are quite healthy and move smoothly. You can see there are no more glowing red, by the way, because it's tissue specific. And here, the motor neuron downregulation of SMN, which is sufficient to reproduce the disease, even in that one very strong. You can see that, for example, this one has a body that is smaller than his head, and we show that it is linked to motor neuron degeneration that lead to muscular atrophy. I will show you later. And we also have progressive loss of motor functions and also progressive development of scoliosis in these animals, exactly as we see in patients. So that was also a big demonstration, very, very interesting. So, and we show that indeed, so in tissue, that the control doesn't have any muscular or motor neuron phenotype. As a muscle knockdown, so no muscle phenotype, no change in the number of motor neurons in the spine. However, when we downregulate SMN1, specifically in motor neurons of this fish, we start to observe a strong muscular atrophy, okay, but not really change in other organs, and a dramatic reduction in the number of motor neurons in the spine, okay, and we show that it was linked to progressive motor neuron degeneration. So it's great just to show you, so the power is just to show you both these new models I created that really excite me to now understand SMA, and I have really now new tools, fantastic tools to investigate the disease, but as well to now involve, to use my chemical genetic approach. I will come back to that right now. But also just to illustrate you the power of these tools and something that you can use not only to model disease and create, as I will show you, models for large scale screen, but also to just study gene functions in your study. Yeah, these models have also interest for ALS, but I will not come back to that. You can ask me a question. I'm already very late on this talk. So now what I'm doing about drug screening. Actually, we're just starting. So just me illustrate you one thing. So I want to show you a third very strong potential and very strong advantage of my tools. Here, for example, to have these animals I show you here that I put in this plate, what I do is I uncross these fish that have one copy of my transgene, the first one you saw. And they're heterozygote for this transgene, okay? So when I uncross them, I got this fish with one wild type, it doesn't get any transgene, two heterozygote, one copy of the transgene, so they will not have strong defect early on, okay? No big motor function defect, however, they will develop the late form of the disease, as you saw. And then a fourth fish, 1 on 4, will have two copies of the disease, it will be glowing red, and it will develop the disease. It is the one you put here. So first to illustrate, this is a strong advantage here because when you make your fish, you can identify your animal before it will develop the disease. And the intensity of the color will tell you how severe will be the disease. So that's very powerful because you can sort that and put that in your plate. What you can't do with mutants. When we work with CRISPR, for example, most of the time, we want early, in zebrafish world, we want an early phenotype. If not, we don't want too much an adult phenotype. We want early phenotype because this is where the fish is useful when we want to work on early on. That means that usually when you have an early phenotype, it doesn't survive until adult. So that means you CRISPR, most of the time you need to maintain them in an heterozygote state. So when you in-cross them, you get the same repetition. However, you have no way to be able to identify the homozygotes that will develop the disease before you do your experiment. So you need to analyze them all, and at the end of your experiment, you need to genotype your fish to know which one was the one that had the disease. Yeah, at the end of the experiment, you should know. But anyways, that's a big drawback when you do disease modeling and screening on disease. Well, again, with my approach, you have that's fantastic, very powerful. However, in my case, I want to go quick and be able to do kind of automatic screen. There is two main limitations, which are actually big. The first is, in reality, when you screen your fish, depending on his position, the intensity he has, even if it is always the same, will look different. So you might, if you go too quickly, you might introduce some of this fish in your screen. You don't want that because this one will look as normal and will introduce a mess in your result. And even me with experience, I still struggle sometimes to do a 100% accuracy in my manual sorting. But on top of that, you may understand that this one I told you will develop the late onset form of the disease, and they have the same genotype as this one. That means that this one also has late onset form of the disease, right? So actually, to get this fish, you need to make this fish, and these fish are those one. Remember this video? So first, the zebrafish is usually adult at 3 months. So first they die around 7 months, so you have a short window to produce your eggs. But second, they have difficulty to mate, all right? And third, they actually don't release a lot of eggs for whatever reason. They produce way less eggs than a wild type, than a control. So this is a big deal because then you struggle to produce enough number of that, and so you have very low number of that, and on top it's hard to manually sort them. It's not hard, but you can mess up, and in a large screen you don't even to mess with one of these larvae. So we are actually working on a third system to try to really optimize our approach for large scale screen. So here was the first model. This is the second model. Oh, yeah, you might ask me why I'm not using that, because actually if people know a bit about the GEL4-US system, you may know that you can maintain the US in a silent state, so you will have a responder line that is healthy. But actually, you cannot really use that because we found out the hard way and that ubiquitous expression of GEL4 is little. So I cannot create a ubiquitous form using that system. And on top of that, we know now that the US sequence is prone for silencing. It's like a magnet for methylation. I think it comes from Drosophila. In Drosophila, they don't have this problem, but in vertebrate, it's what happened. This CPG island repeats in the US attract methylation, and which depend on the cells will attract more or less methylation, and that will lead to a silencing of the expression of your cassette. And what in real world lead is you will have in the same clutch of fish that have different expression of your cassette, so they will not have the same level of knockdown, the same level of the disease. And even if it was not enough, even in the same fish, you don't have this between cell to cell, you have a heterogeneous silencing in different cells. So anyway, it's just too much of heterogeneity when you want to establish a large-scale screen. However, I will not go in detail into that. It can be very powerful approach if you want to do cell-specific study. Not tissue-specific, but cell-specific study. Ask me about that because it's very, very powerful actually, and I don't present that here. We are working on a paper to demonstrate that. Very powerful for cell-specific study. So we are working on the third generation of models to allow really a large-scale screen. So what we do is now we use a Krilox system, and we create a driver fish that express I3 in the tissue, in different tissues. So here, for example, ubiquitous expression of I3 in all the cells of the fish. It's okay. They are just healthy. We have also fish expressing muscle, motor neurons, whole brain, etc. And this fish express also GFP in their lens just to be able to identify them in a clutch So that's a marker because you cannot see I3, right? And we have responder line. So that's an independent line, healthy, perfectly healthy. You can maintain it, easy. And here, a responder line that have our cassette I'm talking about since the beginning, DS-RED and my macroRNA against SMN1. But this cassette is not expressed because silenced behind a BFP cassette stopped by 3 poly-A. And this is ubiquitously expressed. So this fish is fully blue. However, and healthy, because this cassette is not expressed here. However, when you cross these two fish, meaning in presence of CRE, this cassette, this LOXP will recombine and will cut out BFP, okay? And then you will have expression of my DS-RED and macroRNA cassette. A new fish, when you will introduce CRE, your fish will turn from blue to red with green eye, and they will express the macroRNA, and then they will have the disease. So here we are, or how we are, that's the shape we are. We have created heterozygote fish for each transgene, so driver lines, which have one copy of the transgene of ubiquitous expression of I3. They are perfectly healthy, and one and responder line express. We have different one actually, but quite kind of a bunch, but that have one copy of our transgene in their genome, so they are also perfectly healthy. And we show that when we cross these two, we obtain the kind of the same repetition as before with wild type, one which has one copy of the driver transgene, one copy of the responder transgene, and here the fish that will be sick, and this is the only fish that will be red. So first, you cannot mess up with your screen now because only the red fish will be the fish that will be sick, and you don't have different level of expression of your macroRNA because you have just one copy of your transgene. And you can produce as many eggs as you want for a long period of time because this fish can be maintained healthy. But we will not stop here because when we will have homozygote fish for each transgene, we will not hear that yet, but I'm pretty sure it will work, hopefully. When we will make them, 100% of the clutch will have one copy of each transgene, meaning they will all be red and expressing my macroRNA to very similar level, and they will all develop the disease. So here we just have to make this fish, take these animals, and load your plates. So absolutely great to establish your screen. So this is where we are heading to, and not yet there, but and hopefully that will open me the door for SMA for this chemical genetics approach. But just keep in mind that this CRE-LOC system and all tools I presented you can be very useful in new research, and hopefully they will really help you. So finally, for ALS, so we try to be very short. We are trying to apply the very similar principles with my ultimate goal of generating ALS model in the Zebrafish. I will show you why. But just briefly, I'm collaborating with Edo Kabashi in Paris on that project, as well as Professor Naomi Ray in Brisbane. And we are trying to use these tools, but not only, actually. I'm talking about this tool. We're also using gain-of-function approach, also CRISPR. But anyway, so we are using these tools to study the individual and synergetic effect of genes related to ALS. So why introducing these tools in this ALS project, in addition to gain-of-function and CRISPR? First, because we have now data showing that several knockdown of this gene lead to is toxic. So you cannot really, as I showed you, create a model that would be very stable. And some looks like some also trigger genetic compensation, which you don't have with my tool, with the tool I presented to you. So that's very powerful. But also because these tools will allow to create polygenic knockdown. We can now, and we know that most likely all these genes play in the same network. It's a very complex network. So we definitely want to study the gene interactions and the synergetic toxicity of this gene. And using my approach is kind of straightforward. You can chain different macroRNA targeting different genes, and then look at the effect of the combined downregulation of this gene, but not total absence, okay, which probably is little, and it is not reflecting the disease, right? And yeah, obviously the endpoint will be to create a model of ALS that have an early phenotype that we can use in the screen as well and study. And just in terms of pure chemical genetics, because it's kind of the case I made today, I will screen the pool of compound I show you on my SMA model here, and I will find a bunch that aggravate or rescue the phenotype. And if we are successful enough to create additional MND model, as for example of ALS, and we apply the same principle, hopefully they will have phenotypes that we can use in the same screens. Hopefully we'll be able to identify compounds that rescue both disease, okay, and that will help us to identify mechanisms that are implicated in motor neuron disease at large, because probably these drugs will act on downstream mechanisms of motor neuron degenerations. And we will also find specific targets, which also help us to identify upstream mechanisms involving motor neuron degeneration and upstream mechanisms specific to each disease, to understand the specificity of each disease and the shared mechanisms. But that will also obviously help us to try to develop drugs that would also treat each individual MND, and probably try to find drugs that would potentially be beneficial for a large number of types of motor neuron disease. And last but not the least, I'm also a strongly believer that there is strongly link between SMA and ALS, and that SMN1 is involved, SMN1 function is involved in ALS as well. And beyond that, my primary data also show that most of these genes we study that are linked to ALS, meaning when we study them, we got phenotypes, but in my primary data, we got adult phenotype. It's not something that attracts me too much in zebrafish, having adult phenotype. The zebrafish is very attracting when you get early phenotype again. So for these specific genes, my strategy will be to try to create a sensitive background to increase the toxicity of this gene, to create an early onset form of ALS. And for that, I try to create a stable form of SMA which have around 70% reduction in SMN1, so not enough to really create a big mess early on, okay, but strong enough to make the motor neurons and the whole system fragile. And then if you add something more, if you test that will synergize and potentiate the toxicity of our ALS gene, hopefully, and probably will help us to create models that will show an early phenotype. And yeah, that's what we are trying to do. Anyway, so sorry I've been a bit long, I believe. So thank you very much for your time. I would like to thank obviously different supervisors that helped me to reach my goals and helped me to develop my own career and our own group. And I also would like to thank my different collaborators that were implicated in this project, and all the foundations and governmental funds that helped me to move on this research.